Adult female skull and star-child skull, same size. There are 25 major physical differences between these two skulls. I do not have time today to go, my usual presentation is to go over and chronicle those 25 differences so you can see how amazing they are. I'm just going to have to buzz through them today because of the other stuff I have to tell you. If you want to know more, there is an e-book. Go to the starchildproject.com, and there's an e-book that has all of the technical material covered extremely well with photographs and illustrations. For the paper book of the backstory, go to amazon.com. Human mitochondrial DNA contains 13 protein encoding genes and 2 RNA encoding genes. Each mitochondrial DNA has 16,569 nucleotides. It's so small, it's so precise, they really, really know about mitochondrial DNA. Here are the 2 RNA genes, and here are the 13 that are protein encoding. This is a segment of 167 star-child nucleotides on the top line. Now I'm going to show you a blow up in that blue block, so don't freak that you can't really see it very well. This is a relatively small fragment of star-child DNA between, these numbers can be confusing, 1,269 and 1,426 of 16,579, excuse me, that should be 69, nucleotides in human mitochondrial DNA. It shows 17 variations. You have 12 variations here in the close-up block and 5 variations here. This close-up view reveals the way the computer program inserts or deletes as necessary. Now again, that's this block right here, just blown up. Look at the differences. That's the 12 variations right through there. You see them? Between the star child and human in this short, brief area. Now, this you got to get a grip on. This is so cool. Human mitochondrial DNA is very highly conserved from 1,260 to 1,380. That's all we could get on this sheet and make it look halfway decent. But we can go, you know, all 16,000, all up from here to here. You can see 16,000 of these things laid out in these software programs. Every single nucleotide is known and understood in the mitochondrial DNA. So this is just a little fragment. The key here to understand is that this is all of your human haplogroups right here. Human, human, human haplogroups. Human, 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 and Neanderthal and Denisova bone and Denisova tooth down here at the bottom. You know, Neanderthal is fairly different from human. Denisova, the new creature, is even more different, and yet in this area, and in many areas, there are no differences between them and humans. There's certainly no differences in here between any humans at all. This is a highly conserved area of mitochondrial DNA. It's not that there are no changes in humans, you'll see some in a minute, but in this particular area of this particular piece of DNA that we're looking at, there are no differences at all. Because DNA sequencers are so highly conserved, they mutate very little and are virtually the same for all mitochondrial DNA haplogroups. In a segment of human DNA, a fragment, that does not change in any of its many haplotypes, star-child mitochondrial DNA shows 17 variations. It is clearly not human. Now, this really is a touchdown. I hope you can see that. This is as touchdown gets. Yeah, it is. This is the proof we've all been waiting for. I'll show you some more. Now, there are variations in the mitochondrial DNA of human haplogroups, and they are well established in databases. All humans, all of us here, have between 10 and 66. Only 10 and 66 in all 16,000. In all 16,569 nucleotides. You with me? Now, you see that what they look like here? You have one on this haplogroup right here, has one there. The Denisova has differences here, here, here. This haplogroup has one here, this haplogroup has one here, this haplogroup has one here. You see these two red arrows? That's where we're going to look at a little box here. 1,001 Starchild nucleotides in 2 fragments versus 881 human nucleotides in a gene. Okay, that number here gives you the 881, and this one together make up the 1,001. Again, we see close-up of 255 of 881 nucleotides. There are 8 deletions or insertions in between the Starchild, the top line, and the human. And now notice, we're not trying to sandbag you here and find just bad places. Here's a stretch where it's all the same. No changes. Just pretty much done at random, really. But we wanted to make points, and the point we want to make is that what you just are looking at right here, but the big one, covers over here. So where you have 8 variations with the Starchild, you only have 2 here. This one, this haplogroup, and this haplogroup. But with the Starchild, you have 8. Again, not human. Not human. It means its mother wasn't human. It is a pure D alien. Well, excuse me, that's a little strong. It is human-like, but it is not human. I have been cautioned by the geneticists to say, rather than say the inflammatory word alien, to train myself to say human-like rather than alien. But it's not human. This is another random fragment. Our geneticist has dozens. In this one, 545 Starchild nucleotides are compared with 562 human nucleotides in a gene. Excuse me. In this close-up, I went through 150 comparisons and quit marking at 15 variations. Several more are below the green line. You see, it becomes repetitive. Everything he threw up at me, it was again and again and again. It's overwhelming, the evidence here. But it's partial, and it's not going to be accepted because of that. But it is real, and it is legitimate. I wouldn't be here if it wasn't. Late February of last year, 2010, about a year ago, right now, today, as I'm standing talking to you, about a year ago, I provided our geneticist with a Starchild bone sample. He had a valid result in March. After 11 years, the Starchild's nuclear DNA was finally gloriously recovered. This gel sheet shows several fragments. Right there. There it is, folks. TD. Touchdown. Some of the Starchild DNA fragments, this one 342 base pairs long, proved some human Starchild nuclear DNA is not human. If you look down at the bottom, it says no significant similarity found. No significant similarity found means some of the Starchild's nuclear DNA cannot be found in the NIH database, as I just told you. Draw your own conclusions. For reasons why, it also says that, for reasons why, click here. This refers to a list of possible errors in the submission process. Our geneticist obtained the same results with multiple samples, so he has no doubt he did them correctly. Okay? So there it is. Home run, touchdown, we think. But, as usual, the mainstream had an answer for us. Mainstream geneticists dismiss those results because BLAST reports, and that's what those are, BLAST can be fixed by expanding or contracting the search parameters. Unfortunately, that is true. If you want to jimmy the system going in, you can set the parameters so you'll pretty much get back the answer that you want. You can do that. Now, who in their right mind would do it? Certainly our guy didn't do it, but this was one of their ways to try to flush him out. Because when I would argue, they'd say, oh, well, tell us who he is, and then we'll just talk to him about it. And if we tell him who he is, then he's out of a job and he's gone, and we lose him. Can't afford that. Anyway, so he tried other approaches. A spectrophotometer reading for human DNA and Stadchild DNA. This is a test with fairly loose parameters. You can see that the Stadchild is clearly human-like. It wants to be human. It's trying so hard to be human. It really is. You can see it's making a sincere, genuine effort. I want to be human. I'm almost there, but not quite. Now, an electropherogram analysis of human DNA and Stadchild DNA. This is a test with tighter parameters. The Stadchild clearly, this is the beginning and the end, the Stadchild clearly is not entirely human if you go with that test. We know without any doubt that the Stadchild's DNA is not entirely human. It will take a decade or more to chip out its genome piece by piece by piece. It will take only a few months to recover the full genome using 454 technology. The geneticist has done all that he can do to get things to this point. Anything he does from here on is basically repeating himself. He needs to move forward, and so do we. You've seen it. There's really no more to do at the fragment level. There is no more to do. Only action counts. If you can help us make this happen, please do it. This is where we are. This is what we think we can do. This is what we know we can do. We're now in the process of trying to get the money to start the process of sequencing the whole genome, because when you sequence the genome, at the end of that period, it's only about 3 to 4 months. From the time you start, 3 to 4 months, you have an answer. And you have a general idea of what that percentage is going to be, whether it's 15%, 20%, whatever. You can do it within, say, 5 points of error, just recovering the whole genome. And then you have the period of 8 to 12 months where they're going to have to fine-tune it and figure out all of the unknown parts. And then they'll be able to say within a margin of error of 1 or 2% what the difference is. And that difference is going to stick, because anybody that does it again, that's the beauty of DNA. It's the math of biology. It's repeatable again and again. And this is why we win, because we are producing something they can't argue with. There isn't any of this business about nature can do anything. When it comes to DNA, DNA trumps nature. So whatever the DNA says is whatever is going to have to be accepted. And this is what we think it's going to say.